Hello, hello, good evening, everybody. How's things? How is life? Um, excuse me. So tonight we are going to kind of get into the special senses, right? We have been talking about really the nervous system for, the better part of almost 3 weeks, right? We began with chapter 12, right? Just simply looking at nervous tissue, looking at neurons, nerves, what neurons were made up of, how those neurons combined into make entire nerves, and then nerves gave rise to the spinal cord and the brain, etc. Right, and then we went from there, and we looked at some of the different tracts and some of the different highways that these different neurons traveled through to get to different parts of the brain, and then in conjunction with that, we then looked at those individuals in those specific parts of the brain, right? So, right, the purpose of this presentation is to really look at and to understand what some of the special senses of the body are, right? We're going to look into vision, into hearing, and taste, right? We're going to look into these different senses and get a grasp of what some of the different structures that allow us to be able to hear or to be able to see. Right? Before I get into the actual presentation, one thing I wanted to mention on D2L, right now, you undoubtedly have seen the poster, hopefully, anyways. On D2L, but on D2L, in the content section, I have included a Word document where you guys are able to see all of the different structures that I want you to be able to know and identify. Right? I understand over the last few weeks, right, not having class in person, right? As I mentioned, there's a multitude of reasons why. I've conducted, and many instructors have conducted the courses asynchronously, basically recording them like this and posting them, because many of you guys, I know 6 or 7 of you at least have young children, so the idea of having to attend class at this point from your home, being able to listen and actually attend class, I realize that's not feasible, right? So this was kind of the best route I felt to go. So in saying that, right, there's definitely a number of, a lot more kind of talking than normal, right? You guys have the first 2 months of class, right? There was, of a 2.5 hour class, I'd talk for an hour and a half at most, and the last hour and a half, you guys would have the opportunity to, look at the models, kind of study together. I'd come around, answer questions, ask questions, kind of do a little, quiz you on certain things. Obviously, that can't happen, or that didn't happen, right? That's just kind of how things changed thanks to the pandemic we're in, right? So, my whole point in kind of going into this, minute digression is I did a lot more talking about kind of physiology and function of different tissues. Therefore, perhaps that kind of helps you better understand the anatomy, right? Whereas we don't have the models to look at and to help kind of get hands-on and understand things. I felt if I went a little bit more into kind of the pathways and kind of an overall picture, as well as the physiology a little bit, it might help kind of hone in or help you remember some of the structures a little bit more so. But on D2L, right, I will include a Word doc that has the different, structures, right, the different nerve structures, the different neuron structures from the axon to the axon hillock to the soma, the dendrites, right, to in the brain where the brain stem and the medulla and the midbrain, all those things, right? I'll have on that list all of the different structures and regions and parts that I want you guys to know, right? And then the images that I will pull to create the exam, you guys have those, right? The images that you have on the PowerPoints, those are the exact images I will be utilizing. Right? So, right, make sure things are organized, because remember, the exam is open book. Right? Open book, you can use the PowerPoints, you can use your book, notes, whatever it happens to be. Right? Have the PowerPoints out, have them accessible. Right? You can use them. Right? Have the PowerPoints cataloged, or have them kind of separated, so you know if a question is on the spinal cord, you will not have time to look at every single question, right? But you know what? If you can quickly know exactly what page or where one particular structure is on, you should be able to quickly flip to it if need be. If need be, right? So, anyways, I wanted to throw that out there before I forgot. So, on with what we're going to be covering this evening. Again, that's the special senses, right? Now, the first one we're going to be taking a look at, right, is our sense of smell, right? Our sense of smell. That's something called olfaction. Somebody, God bless them, big fancy term, right? Instead of smell, it's called olfaction. Now, if you remember last class, we talked about something called the olfactory nerve. That was your first cranial nerve. Or your first cranial nerve, that's what allowed your sense of smell to transmit from your nasal cavity up through the olfactory neurons, and it synapsed with the olfactory nerve. Right here. Right, how we mentioned last week, there's two different parts to the olfactory nerve. There's the olfactory tract. The olfactory tract, that's kind of this part in through here, all down and through here. The olfactory bulb, that's kind of the end part, the end portion, the big bulbous region at the end of the nerve. And so when we see, right, this is what it kind of looks like. Right here's the olfactory nerve, right, or parts of it, specifically the olfactory bulb. But we can see how, right, if you remember, back to the ethmoid bone, right, going back a month ago at least, when we think about the ethmoid bone, there was the cribriform plate, there was the olfactory foramina. Right? If you remember that the only way to see this was from an internal view of the skull, right? Pop the skull cap off, look inside of the skull, and look up towards the front, almost about where the frontal bone would be. And there was this little, not very big, right? The area is not any bigger than, it's oblong, right? It's smaller than a marble. Right? It's not round like a marble, but it's kind of drawn out. Anyways, in that area, there's tiny little holes called olfactory foramina. Those are the holes which these olfactory neurons can actually enter up into the olfactory bulb. Right, you can see a couple things. Olfactory gland, mucus layer. Yeah, think of it this way. Take a thing of sugar, dump it on your table. Just leave it be. Right, don't touch it, just leave it be. Go do something for a few minutes, come back, look again, you're like, sugar's still there. I have no idea what's going on. In your nasal cavity, right, one of the things that actually happens, right, if you were to take that sugar that I just said to dump on your countertop, leave it for a few minutes. After a few minutes, you realize, nothing's happening. Then take some water and dump the water onto that sugar. What happens to the sugar then? Oh, some of it starts to dissolve. Not all of it. If you keep dumping more and more and more water, the sugar would all dissolve. But that's how your sense of smell works. Right, the olfactory gland here produces a huge amount of mucus. Right down through here. Little tiny particles, which are emitted from, right, whatever it is that you're eating. Right, well, whatever it is that you're eating, whatever is emanating the odor, right, hopefully it's not, the remnants of Taco Bell, right, that's enough to do you in, right. But whatever it is that's emitting some sort of odor, those particles get lodged in the mucus. And just like those sugar particles, which if you dumped a bunch of water on them, they would dissolve. That exact same thing happens to these odorant molecules. They begin to dissolve, and the rendering chemicals comes into contact with these little dendrites. Excuse me. Right? So when that happens, right, depending on what chemical it is, right, it sends that impulse up the neuron, eventually up into the olfactory bulb. Right. You can see basal cell, right, this guy here. Basal cells, yeah, think of basal cells as being like baby cells. Right, babies can grow up to be an adult. Babies can grow up to be a number of things. Basal cells, right, they can't grow up to be a number of things, but they can grow up, meaning your olfactory epithelium, right, which is all of this, is unique, right? And the fact that there's a number of nervous tissues, right? There's a number of nervous cells which exist throughout the olfactory epithelium, and they can regenerate themselves, right, because of the presence of these basal cells, and that's unique. Right, that is very atypical for nervous tissue. Right, but that can happen with these olfactory receptors, these olfactory neurons in the epithelium. So here's just another view, right? You can kind of see this olfactory gland here. You can see from here down all the olfactory epithelium, right, all that through there, right? Same thing we're looking at, but just of an actual, right, of the actual nasal cavity, right, from an actual person, an actual individual. This is what it looks like, right? So just like within the olfactory gland, producing that mucus would then get ejected, emitted, secreted out here, so therefore it would cover the olfactory cilia. That odorant molecule would come along, it would get caught in this mucus, it would dissolve, and it would stimulate these olfactory neurons, again, which then send that impulse up into the olfactory bulb. Cool. Your sense of taste works incredibly similar to that. Right? Incredibly similar. In the sense that, right, let's imagine hypothetically you didn't have saliva, you wouldn't taste stuff. Right? If there was no moisture in your mouth, right, you wouldn't be able to taste food. So a couple of things. We see papillae, right? Fungiform, villiform, valate, circumvallate. So we see these kind of bumps, right, in through here. These little ridges on the side, right? This big, large thing, right? These are the lingual tonsils here, palatine tonsils here, epiglottis. Now, one thing is to not get like super confused as to how we're looking at this. This is a superior view, right? So, for instance, this is all in like the very back of the tongue, right? When you open your mouth and look into a mirror, right, you pretty much only see from like here forward, right? You don't see any of this stuff, right? Especially this. Right? This here, right? Think of it this way, kind of how this particular image is. If you were looking into someone's mouth, right, this essentially would be kind of looking, or here is where they're kind of going down into the throat. Follow me? Right, like if you were to take a thing of food, right, have a whatever piece of candy, put it on your tongue. When you swallow it, right, when you swallow it, that's the only point that it gets back to this region, back to the lingual tonsil, and especially down to the epiglottis. The epiglottis, that's at the area, right? Again, the area of like the Adam's apple in males. Right, that's how low the epiglottis is. So that's where this particular structure is found. So, as far as the papillae, right, or papillae, however you want to pronounce it, you can see there's a few different forms. The filiform. Filiform, by far and away the most abundant. Right, they are very small and spread throughout the tongue. Right, really, they add much more of a kind of an abrasive force. Right, they don't have any taste buds, but they do kind of help to adhere to the food that you're chewing and having in your mouth, and it helps to kind of turbulate the food so it better mixes with the saliva. The fungiform papillae, right, the foliate papillae, they both contain taste buds, right, and you can see they're far less abundant. Right, the fungiform are much larger. Right, they're almost kind of mushroom shaped, hence fungiform. Right. If you've ever had a kind of red bumps on your tongue, kind of spotted throughout the tongue, right, like kind of a red area, which it's very small, it's kind of pinpoint, but it's kind of irritated, right, and sore at the touch. Most likely, those are the fungiform papillae. Right. Lastly, the vallet papillae, you may also see them called circumvallate papillae. They're massively big, huge, right? Literally, there's maybe a dozen, 15-ish or so, right, on the posterior aspect of the tongue. Right. And they actually are they actually produce a saliva. Excuse me. They actually secrete something called lingual lipase. Right. That's one of the secretions which begins to help break down of fats. Right. Anyways, those are the circumvallate papillae. And here, just another kind of image that we're going to see here. Just like I mentioned before, right, the function of taste is very, very similar to that of our sense of smell. Right? These taste cells, right, let's imagine some food particle comes along, dissolves in the saliva, the rendering chemical comes into contact with the taste cell, or with that, that tasting receptor. Taste and gustatory, same thing. Right? Gustatory is your sense of taste. So that particle of food comes along, comes into contact with the taste cell, stimulates the cell, and it generates an action potential and travels back to the gustatory centers of the brain. Perfect. See right through here. One thing, it's almost kind of, well, not kind of, a little bit of a review from, 2 months ago. What type of epithelium? I'm going to ask the question to give you about 10 seconds to think about it. What kind of epithelium lines the inner portion of the mouth, right, as well as the tongue? Take about 10 seconds to think about it. Right. Now, yes, I realize it was on the previous slide, it was on this slide, but stratified squamous epithelium. Remember, stratified squamous epithelium that functions to kind of help, it's in those areas to resist abrasive frictional forces. Right, inside the mouth, that's weird. And food, right? The food that you eat is very abrasive. Right? Think of a thing of chips, nuts, hard, rustic bread, Captain Crunch cereal. Good Lord, I think swishing around a bag of glass shards would do less damage than Captain Crunch, as delicious as it is. That stuff is brutal. Right? But even then, right, stratified squamous epithelium still gets kind of irritated and worn away. But that's its function. That's why it exists inside of the mouth, right? Later in 122, you guys will learn about a few different things, especially like in the GI tract, right, or the gastrointestinal tract. our body's able to adapt or modify what epitheliums are in certain areas. It's not normal, it's abnormal, right? But people who have, let's say, GERD, right, gastroesophageal reflux disease, right? Think of acid reflux, right? Heartburn, but like, right, heartburn and steroids. Their esophagus, right, is lined with stratified squamous epithelium, but it's continuously coming into contact with stomach acid, hence the reason they get the heartburn. That then forces a change in that layer of epithelium lining the esophagus. It's called Barrett's esophagus, right? You guys will talk about that in 122, and that's one of the mechanisms or one of the ways that our body can actually kind of protect itself, right? It's an abnormal. It's a normal response, but it's an abnormal tissue, right? Because stratified squamous is what lines the esophagus, right? But in the presence of Barrett's esophagus, or what it changes to, is columnar epithelium, right? So, anyways, going from our sense of taste now, so well, sense of vision. I'm sorry. Here, taste, olfaction. Yeah, they are exceedingly quick to get through. Right? There's not many when it comes down to it. There are very few structures, especially compared to vision. Right? There are very few structures which are just associated with our sense of taste and our sense of smell. Right? That's not to say that there are simple senses. Right? That's not to say that they are, it's not to say that they aren't complex or that they don't project to many areas of the brain, because they do. Right? Our sense of smell is hugely important to our sympathetic response to a myriad of different events. But just as far as from a structure standpoint, there's just not that many structures which help us to achieve our sense of smell. Right? It's much different than the eye. Right? The eye, and really part of the reason why the eye has so many different structures involved with it is, we'll look here in a few slides, but in order to be able to see clearly, right, there is a specific pinpoint area inside of the eye that the light has to focus on. If it doesn't, you don't get a clear, crisp image. Right? Just like, many of you will never understand this, and it breaks my heart, but it makes me happy for you as well. Some of you will understand this, unfortunately. But old school cell phones. Right, nowadays, you pull your cell phone up and it will always be in focus. But when they first came out, oh my God, sometimes you had to mess around for 30 seconds, 2 minutes to get your phone into focus. And why? That's because the lens inside of the phone, all of that had to work in unison in order to get that image in focus. Right? Same idea with our eye. Because it's dependent upon the light entering into the eye, there are so many structures which are available and so many structures which are necessary in order to actually get that light to focus on that specific part of the eye. Right? So, before we get into the eye, before we get into looking at what's the specific part or structure that these light rays focus on, first we're just going to look at some of the more protective things of the eye. Right? Okay, eyelashes, right? Eyebrow, okay. The black of the eye, and that's something called the pupil. This is literally a hole. Like if I could take off, right, this outer covering of the eye, this would just be an open hole into the eyeball. Right? Surrounding it, right, the colored portion of the eye, that's something called the iris. That's something called the iris, this colored portion of the eye. Here, in the medialmost portion of the eye, something called the lacrimal caruncle. The lacrimal caruncle. This is involved, well, in a minute we'll look at, but lacrimation or tear production, fluid production. Right, this is an ongoing thing. It's not like you only produce tears when you cry, right, or when you're out of, I don't know, the last sip of a milkshake. That often brings a tear to my eye, right? But our something called the lacrimal gland, it's continuously producing tears because something has to keep our eye lubricated, right? Something has to keep our eye moist. And the reason why that is, overlying the white part of the eye, so the white part of the eye is the sclera. Overlying that is something called the conjunctiva. It's something called the conjunctiva. The conjunctiva, it's a membrane, right? It's a membrane which requires fluid. It's a membrane which requires nutrients, right? Just like if your skin were, if your skin was dry, you put lotion on it. Your skin is a cutaneous membrane. Right? The conjunctiva, okay, it's a mucus-based membrane, but it's still a membrane. If it dries out, that's when we start to get kind of red eye. Right? You know what I'm saying? Like your eyes are kind of red and bloodshot. You're like, oh, it just feels like there's like sand in my eye. Yeah, it's because it's dry. Right? If you're trying to, as you're trying to blink, as your palpebrae or your eyelids, right, as they're closing, we're just kind of scraping along the conjunctiva, right? If that happens enough, the conjunctiva gets irritated. If there's some sort of infection and whatnot, that's where something called conjunctivitis can take place and develop. So here's a look at kind of the inner part of the eye. Now here's where we start to get into some of the more specific structures and features of the eye from an internal aspect, right? So, one of the things that we're going to see, and actually let me back up for a second, I forgot to point this one thing out. So as I mentioned, kind of the whitish part of the eye, that's the sclera. Cool. Over top of the iris and over top of the pupil is something called the cornea. Something called the cornea. If you've ever put contact lenses in your eye or ever seen somebody do it, right, the cornea, that's what you're putting the contact lens on. That is the cornea. Right, so again, if you've done it yourself, seen a movie, seen somebody put in contact lenses, that's what that is, right? That is the actual cornea. Again, that's what we're seeing right here. Right? This is the cornea. Cool. So, now granted, this eye is shut, so no light is getting in. Light is stopping here. But if the eye was open, all of these structures from the pupil to the lens to the iris to the cornea to the what's called the humors, right? They're not listed. We'll talk about them in a minute. But a bunch of liquid, right, inside of the eye. All of them function to get this light focused on one specific part of the eyeball, because that one specific part is what gives us an image which is actually in focus. Right? So, this here, the lens. The lens is, think of like an oblong marble. Is incredibly hard. Right? Like if you were to take the lens of a person, animal, sheep, cow, whatever it is, you wouldn't really even know it's a lens. It's incredibly hard. Okay? And what happens? A very small amount of distortion to the lens can cause a huge change. Right? Think of it this way, right? If you are looking at, if somebody's 4 feet in front of you, right? If you're staring at them, if you put your thumb up in front of their face, and if they're only 4 feet away, you can still see most of their face, right? But if there's something that's like, I don't know, 200 feet off, or 200 yards off, and if you put your thumb up in front of it, It could be a building, or it could be a car that's 100 yards away. But if you've put your thumb up, it's blocked the entire car. You can't see any of the car. Right? But yet, the person that was only 4 feet in front, that was only 4 feet away, that person's way smaller than the car, and when you put your thumb up, it only blocked maybe just their nose or the middle part of their face. Right? So the whole point is, right, how the lens is, the smallest change in the lens drastically impacts, right, the image, right? Just like that car that's 200 yards off. If I just take my thumb and move it the tiniest little bit, I can now block the entire image of that car. Right, so that's why, again, there are these muscles right here. They're called ciliary muscles. The ciliary body, they're identified here in the next slide or the slide thereafter. Right, but these are the muscles which are responsible for pulling on that lens. A couple other things that we're going to take a look at here. When it's involved with movement of the eye, right, the actual eyeball itself, there's a few muscles. This muscle here, something called the superior rectus. Superior rectus. Alright, so the superior rectus is going to pull the eye superiorward, so up. So look up right now. Okay, cool. That was the superior rectus that was achieving that. Now look down. Right, pretend like you're in class and you're going to send a text. See it goes down into your lap. Right, that's the inferior rectus, which is doing that. Now, if I said, hey, look, look cross-eyed. Right, look, or well, let's say if you looked down and in. Right, as if you were trying to look at your chin. That's a muscle called the superior oblique. Right. We don't actually see that here. The next image, next slide, I believe, we're going to see an image of the superior oblique, but it kind of comes around at an angle. Right. There's also a muscle called the inferior oblique. You can see part of it here. Right. But the inferior oblique, again, it's going to help us look up and out. But like I said, with the obliques, we're going to look at here in just a moment. This here, a moment ago I talked about this lacrimal caruncle. Right. And I mentioned how our eyes are constantly needing to be moisturized, right? Our conjunctiva, that mucus-based membrane which lines over top of the eyeball, right? If it dries out, it can become incredibly irritated, and it almost feels like you're trying to blink with an eye full of sand. Right? So there has to be something in order to produce the necessary lacrimation or tears, and there is. It's something called the lacrimal gland. That's this guy right here. Cool. So from the lacrimal gland, we now have what are called lacrimal ducts. The lacrimal ducts release the lacrimal fluid, and that lacrimal fluid then basically essentially coats the eyeball. And that's why we blink. Right? As you blink, that helps disperse that lacrimation or those tears around and across the eyeball. That's part of the reason why we blink so often. We blink so frequently. Right? Especially if it's, windy out. Yeah, reflex, but the other part of it is because of how quickly that lacrimation or those tears are evaporating. So how does it work? Alright, so as the lacrimation comes across the eyeball, right from the lacrimal gland, the lacrimal duct comes across the eyeball, and now it's time to, do something. Well, if it's too much, alright, it can run down the cheek. If you just watch a sad movie, Steel Magnolias, kills me every time. But let's say if you're not crying too much, right? Let's just say you're producing a normal amount of tears, or even if you are crying, it doesn't matter, right? If you've ever noticed, like when you're crying, well, your nose starts to run. It's actually not, nasal fluid. Right? It's actually tears. Right? So you're kind of like your quote unquote your snot is actually your tears coming out, right? And this is the reason why. Lacrimal fluid, as it comes across the eyeball, you can see there's two ways. Here and here. There's the inferior and superior lacrimal canaliculi. Here it says canaliculus, that's singular. Canaliculi is plural, right? But that dumps into what's called the lacrimal sac. Right? That dumps into what's called the lacrimal sac. And that then dumps into what's called the nasolacrimal duct, which is what then dumps down into the nose, down into the nasal cavity. Right? So that's part of the ways. That's part of the way that your eyes get rid of the excess tears. Right? Is to divert them into the lacrimal sac, from the lacrimal sac down to the nasolacrimal duct, then from the nasolacrimal duct into the nasal cavity, right? And then eventually out through the nasal cavity. Cool. So here we see this right here. This is the exact order we had just talked about. All right. Now, continuing on with the actual kind of eye itself, right? Here's where we start to get into, right, the actual focusing of the light. Right, when it comes in, think about it this way. Let me get this off here. Stop. Sorry. Okay. So we have the cornea, the humor, pupil, lens. It's all vitreous humor back in through here. Okay, so we have numerous, everything I just mentioned. Those are all things which can refract light. With the exception of the pupil, but the pupil can regulate how much light gets in. When I say refract light, if you've ever been on a fishing boat, or a boat, you don't have to fish. I've been on boats, I don't fish. Right? Or you've been by a pool, doesn't matter. Been on a floaty. If you ever stuck your arm into the water and you're like, oh my god, my arm just like broke off. My arm is like 3 feet off to the side. Right? That's the reason why that happens, is because water refracts light. It bends light. Well, remember how I mentioned? There's one specific part of the eyeball that we want that image to get to. Something called the macula lutea or the macula densa. Fovea centralis, this is a specific part within the macula lutea. This is the area of kind of just, 4K, like crisp, clear, beautiful, perfect image. That's the fovea centralis. So if we work our way through, great, light. First, that light has to travel through the cornea. Awesome. Travels through the cornea. The first thing I'm going to enter into is something called the anterior chamber. Okay, there's just the different parts of the eye, or different areas of the eye, are just kind of broken up into different rooms, if you will. But instead of rooms, they're just called chambers. So the anterior chamber. The anterior chamber is full of something called aqueous humor. Aqueous humor is just aqueous liquid, right? It is very, very liquidy, hence aqueous, right? It's very watery. From the anterior chamber, that light travels through the pupil, and it hits the lens. Awesome. Here's the lens. From there, as long as you don't have some sort of issue with your lens, you don't have an astigmatism, which I'm going to try to draw. Let's imagine that's a normal shaped lens. It's actually not far off, right, because they're not circles. An astigmatism would be, let's say, a lens that kind of looked like this, right? It is misshapen. Right? So this light, as light went through this lens here, it would then focus it, let's say, to the specific spot. As the light goes through here, well, that may focus it way off target. Right? We'll look at some of the different options in a minute as far as like people who are farsighted, nearsighted, those who have glasses. We'll look at those in a second. Or in a few minutes, not in a second. But, anyways, from the lens, that light eventually ends up to the fovea centralis, or at least that is the end goal. That's what we want to happen. Up here, when I mention these muscles which pull on the iris is the color portion. That's the color part of the eye. Right, look in your eye. Oh, my eyes are blue, they're brown, they're green, they're whatever color they are. Right, that is the iris. The ciliary body. Right, that's what actually pulls upon. Right, that's what actually pulls upon a lens to get it to change shape. Now, there's specific parts. So when we say ciliary body, we see, right, there's ciliary muscle, and there's ciliary process. Okay. So do you remember how we talked about cranial nerve 3? Cranial nerve 3 dealt with the vasodilation, dealt with dilation of the pupil. Right, it helped regulate pupil size. Right, the specific muscle which does that is, in part, the ciliary muscle. Right, because what happens is the ciliary muscle, this right here, attaches itself to the iris. As it contracts, it pulls the iris this way. Right, so the iris gets wider. As the iris gets wider, so does the pupil. Right, the pupil gets wider, or it dilates. Therefore, it allows more light into the eyeball. Right, because the more light that gets into the eye, the more visual information, the more visual sense we are able to receive and transmit back to the brain. Right, that's why we talk about it's your sympathetic nervous system which dilates the pupil, makes the pupil nice and big. Right, because when the pupil's nice and big, that means we can allow a great deal of light into the actual eye. Cool. Now, as we get further and further into the eye, there's different layers of the eyeball. Right? The innermost layer is something called the retina. That is something called the retina. The retina is the layer which has all the photoreceptors. Like, I don't know what that means. Photoreceptors. Yeah, touch something. Doesn't matter what. Just touch something. As you touch that, you can feel it. Why? Because the little touch receptors in your finger got stimulated and lit up parts. Lit up the corresponding part of the brain and say, hey, you're touching something with your finger. Photoreceptors, they don't detect touch, they detect light waves. Right, so when light waves come in contact with them, that's what stimulates them. Right, we're not going to go into that whole process and how and what chemicals. Nope, no thank you. Right, we won't be going into that. But that's what stimulates these photoreceptors, is actual visible light rays. Cool. So from the retina, right, the retina being the innermost, if we work our way back out, is something called the choroid. The choroid, and that'd be the middle layer. The choroid, that's just where essentially by and large, the blood vessels, right, the blood supply of the eye exists. Right, and the very outermost is the sclera. Think of the sclera as kind of being like the skeleton. Right, think of the history lesson, the Revolutionary War. Right? Don't fire till you see the whites of their eyes. Oh, this was the whites of their eyes. This is what they're talking about. Right, the sclera. And this gives the eye its kind of round. When you draw an eye, you draw it round. Why is it round? It's round because of the sclera. Right, just like our bodies. Why are we shaped the way that we are? Because of our skeleton. Everything hangs off of our skeleton. Our muscles, our organs, etc. all hang off of our organs, or off of our skeleton. That's why we have the appearance that we do. This, what's called the vitreous chamber, kind of this whole, like literally this whole back part of the eye. That's the vitreous chamber. That's something that is filled with vitreous fluid, what's called vitreous humor or vitreous body. Right, again, this whole back part through here. All right. One other thing that we want to look at, if I look right here, cool. The second cranial nerve enters into the eyeball. Right. Or really, I guess vice versa, so it would be the opposite. Right? The visual receptors, which those photoreceptors within the retina, all travel and combine and accumulate and enter into the optic nerve. And they go from the optic nerve, travel back to the brain, specifically the occipital lobe. And that's the question. I'll give you about 10 seconds to think about it. Look at this spot right here. Right. Right here. This is something called the blind spot. This is where no vision is associated with this area. No vision can be generated from this area. Take 10 seconds to just think about why that might be. So recall, right, remember, in order for a receptor, or in order for you to be able to see something, right, that light has to come into contact with a photoreceptor. Alright. This area here, where the optic nerve, where the blood vessels are entering and exiting the eye, if there's a bunch of blood vessels entering and exiting the eye, can there be any sort of photoreceptor in this specific area? No, there can't be. I can't have both blood vessels and photoreceptors existing in the exact same spot within the body. It's impossible. But you can have two things in the same spot at once. So therefore, because of the fact that there are blood vessels, that means there are no photoreceptors. And because there are no photoreceptors, that area cannot generate a picture. Hence the reason why it's called the blind spot. Or you may also see it called the optic disc. Either one would work fine. Right? Either one will work fine. So here we can start to see a couple of other muscles. Right? The superior rectus, we looked at that previously. Right? That's where the muscle that looked directly upwards or superiorwards. We looked at the inferior rectus, the obliques. Now here's where we are going to look at some more of the recti. Right? The medial rectus and the lateral rectus. Right? The lateral rectus. If you recall, the lateral rectus, right here. The lateral rectus. This is going to pull the eye laterally. So, for instance, talking about your right eye. If we talk about your right eye, Take your right eye and look right. That's the lateral rectus. All right. The medial rectus let's go here. If we're still talking about the right eye, that takes the right eye and pulls it inwards, so you're looking to the left. So, if you are looking to the left, which recti is functioning on the left eye if you are looking to the left? The lateral rectus. If I keep looking to the left, what recti, or what rectus, is working on the right eye if I'm looking left. The medial rectus. Right? So it's essentially when you're looking left and right, it's the opposite rectus for that particular muscle. Right? Lateral rectus, again, that is cranial nerve 6, the abducens nerve, which controls that. The medial rectus, for this guy here, right, that's cranial nerve 3. It's the oculomotor nerve. So, here's just a little bit closer of a kind of a up close and personal, a little better of an image or better of a view. Right, you can see a little bit more of the ciliary muscle itself and how it kind of goes up and helps to control and regulate part of the size of the pupil, right, through the iris. We can see the ciliary process, how it comes and actually attaches itself to the lens. One of the other things that we couldn't see before, there we go. So you see sclerovenous sinus, anterior ciliary vein. Yeah, I mean there's small veins within the eye. The eye is very vascular. It is immensely vascular. Right? That's part of the problem with uncontrolled diabetics, for instance. Right? Even the blood vessels of the retina, very small, very delicate. Right? Uncontrolled diabetics with high blood glucose or blood sugar. The problem with that is that just wreaks havoc on your blood vessels, especially the fragile blood vessels of the eyeball. So because of the elevated blood glucose, the blood vessels suffer, they become damaged, and those vessels which are more susceptible, those vessels which are more sensitive and fragile, are the ones which die first, and oftentimes those are the ones in the eyeball. But here, something called the canal of Schlemm, right? Canal of Schlemm, sclerovenous sinus. Right here, right? Do we see how it kind of has this arrow here? Yeah. So, in the anterior and posterior chamber, remember this is full of aqueous humor. I'm just going to draw AQ. Yeah, that's a Q. Good Lord. There we go. That's not any better. Let's take a tool. I'll live with it. Aqueous humor. Right, it's all fluid. Hell with that. We'll just draw water. Draw a wave. There we go. Perfect. Water. Aqueous humor in the anterior chamber as well as the posterior chamber. This stuff's constantly produced. It's filtered. It is kind of recycled via these ciliary veins. But how it gets there is it travels through the canal of Schlemm, or the sclerovenous sinus. This is important. Right? People who have, if you've ever heard of glaucoma, this is one of the more common reasons for people to develop glaucoma. Glaucoma, there is a buildup of pressure inside of the eyeball. Alright, let's back up for a second. If there's a buildup of pressure inside of the eye, well, if I'm building up pressure inside of the eye, what does that mean? That means the eye is going to try to expand. Right, I'm building pressure. The problem with that is take your finger and pinch it. starts to get painful. If you were going to keep on holding it, you can let it go. That same thing happens to the retina. Right? That pressure literally begins to compress the retina. Right? That pressure compresses the retina, which, again, the retina is very delicate, sensitive tissue, and eventually it begins to cause damage. Right? That's why glaucoma is, right, the leading cause of blindness worldwide. Right? Because of the increased pressure, because of the increased pressure within the eye, that can damage the retina. Part of the reason why that is, or the biggest culprit, is because you can't have this evacuation of the humor. Right? This fluid, this humor, the aqueous humor is just constantly being produced. It's got to have a way to get recycled and filtered out. But if it can't exit and get recycled, it just remains in here. Well, we just keep producing more, and now we can see how that's a problem. Okay. All right. The same thing right here, right, with the optic disc or the blind spot, where you can see how all of these different photoreceptors enter into the optic nerve. All of them. Right, but because of that, this area right here, there is no actual photoreceptor. So because there are no photoreceptors, it cannot generate an image. Hence the reason why it's the optic disc. So this here, right, when we look inside of the eyeball, recall how I mentioned light comes in the eye and eventually gets, boom, right here. Focuses on the fovea centralis. Awesome. That's the area of like crisp visual acuity, perfect, just crystal clear image. That's what we're looking at right here. Right? If a physician, right, has ever looked inside of your eye with an ophthalmoscope, right, this little tool they use to look inside your eyeball, this is what they would see. This is what they would want to see. Where they see this kind of dense area here. Right, this is the fovea centralis, optic disc. Right, and they see this kind of network of vessels throughout the eye. It looks really, it looks just like this. It's very cool, actually. It's very cool. But right here, right, this spot right here, this is where those light rays, this right here is where you want that light to focus. That's why there's the aqueous humor. That's why there's the vitreous humor. That's why there's the lens and the cornea and the iris. That's why all of these structures exist. Because, just like I mentioned, as light travels, it can hit the cornea or the fluid, right, and it bends. Right, just like I mentioned, if you're on a boat or you're on a floaty in a pool, just take your arm and stick it into the water. It looks like your arm is separated from your body. Right? But it hasn't. Right? It's just the fact that that light is being refracted or bent along the water. Alright. So again, this here, right, how I mentioned, if you actually impact how much light gets in, that's one of the main ways we can alter what it is that we focus on. Right? So when we kind of carry on with that, right, depending on where the object is, whether it's far or whether it's close, right, you can see when it's a very distant object, these zonular fibers are taut. Yeah, I mean, there's the lens is being pulled upon. So when that happens, that helps to focus that light onto the fovea centralis, right here. Now you can see the zonular fibers loose, ciliary muscles contracted. Well, yeah. When that happens, when they're loose, right, the lens kind of remains, not fat. It never changes shape, that much, or in a very small amount. But as I mentioned, it only needs to change a very small amount, right? Because that very small change drastically impacts what it is that we actually see. Right? So when we go from, again, a distant object, right off here. Again, the lens, it kind of thins out. As it's closer, it stays fatter. Right? This is something called accommodation. There's a process called accommodation. Let's say if you were to hold your finger out at whatever. Just hold your arm out, hold your finger up, stretch your finger, and then kind of quickly bring that finger to your nose, like to within 4 inches of your face. Right? Not quickly, but just bring it to within 4 inches of your face. You can keep focus on that finger the entire time. Right? And just when you do that, do it 3, 4, 5 times. Just pay attention to all of the different objects in the background. How out of focus they become. Right? That's because as you're focusing on that finger, right, the lens is having to change shape in order to accommodate the movement of that finger. Right? So because of that, that's why it starts to impact, oh my gosh, all of a sudden, as I'm doing it, well, as my arms outreach 2, 3 feet, I only see one laptop. But as I hold my finger only 4 inches from my face, now I see 2 very distinct laptops. Right? So it's all about kind of that movement of the lens and what you're able to see and focus upon. When there is issues or problem with focusing on the image, this is what it looks like. Because it all has to do with, right here's the sweet spot. That's where I want it to end up. Cool. Alright. Well, what happens if somebody is nearsighted? Alright, that means, nearsighted. I can see things pretty far, pretty well close up. Cool. Well, in a person who is nearsighted, what actually happens? So instead of the sweet spot being right here within the retina, let's back up. Right? Don't ever just think, oh, it's just one light wave bouncing into the eyeball off the object you're seeing. Not even close. There's thousands. Right? Like every little bit of what you're seeing, right? Every little bit of the image. Thousands of light waves being, entering into the eye all at once. Right? So here we'll simplify it and keep it at 3. But these 3 light rays, or light waves, are what are going to give you the image of my whatever. There's a thing of Gorilla Glue on my desk. Right? So let's just say in order for me to see that image of Gorilla Glue, there's 3 light waves which are carrying that image into my eyeball. In order for my body, my eye, or my brain to be able to put that image to use and be able to put that image together, those 3 light waves have to be focused on the retina. If they're not, that's where the image looks fuzzy. So in the instance of nearsightedness, right, that's where the actual, right, there's an issue with the lens, there's an issue with the cornea, there's an issue with the humor, there's an issue with the humor, right, let's say the amount of it, for instance. So because of that, the image actually focuses prior to it reaching the retina. Right, so as they, let's say here's the eye, right here's the sweet spot. Well, as those light rays come in, you want them to focus right there on the fovea centralis. In the instance of nearsightedness, what happens is as those light rays come in, they focus prematurely. Uh-oh, they focus before the fovea centralis. Well, they continue to diverge. So you can see how right there is where they've come into image, or come into focus. As they continue onwards, oops, now they're out of focus again. Hence the reason why, right, people who are nearsighted or farsighted, right, hence the reason why people have vision difficulty. All right. Now, same idea, same concept with farsightedness. Right, let's say if somebody is farsighted. Sorry, one more eraser. Go. Farsighted. What's happening? Well, as light rays come in, you can see how, as they come in, they don't actually focus on each other until after, or until past that sweet spot, until they're already past the fovea centralis. Right. So hence the reason that's why these people have a difficulty seeing things which are which are close up. They are able to see things which are further away. So either way, right, in either instance, right, these people can wear glasses, contacts, whatever happens to be, then it helps to correct the image, right? Just think of it. How I mentioned before, as the light comes in, there's the cornea, right? There's the aqueous humor, there's the lens, there's the vitreous humor. All these things help to focus the light ray on the fovea centralis. When you wear glasses or contacts, it's just adding another layer. It's just adding another line, if you will, another line of defense, if you want to call it that, which is able to refract the light. So therefore, the idea is that when you have that added, another line of defense or another line of refraction, we're able to refract that light so it actually focuses on the fovea centralis. Okay? Cool. Alright, so here, this is where we see our kind of how is it our visual information gets from what we see into our minds as to what it is that we are actually seeing, right? How is it that I know I'm looking at this tub of Gorilla Glue? Or how is it that I know I'm looking at a flashlight, for instance? Right, so our eyeballs. This guy, let's imagine here and here. Right, so we look at the eye, I want to kind of just draw the different tracts. Right, in a nutshell. Right, but from the eye, right, part of what goes on is certain parts of the visual sensation from the right eye. Part of it goes to the left occipital lobe. Part of it goes to the right occipital lobe. And vice versa, on the left eye. From the left eye, part of the visual information on the left eye. So for instance, the right visual field goes back to the right occipital lobe. The left visual field goes to the left occipital lobe. All right, we're going to look at an image of that in a second. But that's part of how we are able to kind of render the image of what we're seeing. But before it gets to the occipital lobe, we can see how there's kind of a pretty substantial amount of neurons which are associated to our sense of vision. Right? All these different highway tracks. Right here. So when we look at the eye, right, we just see a little nub of the optic nerve here and here. Right? The chiasm, that's like the crossing over. That's the X, if you will. The tract is really the biggest, longest, part of it right through here. Optic tract. We see the lateral geniculate, optic radiation. Right? Optic radiation, that's kind of like the turnpike, right, if you will. Right? Leading up to that, optic tract of the thalamus. Remember, through your thalamus, right, every one of your senses except for your sense of smell projects to the thalamus, right? Your thalamus, that is the kind of the gate to the higher brain functions, to the various centers of the brain, right? So, for instance, if you were sensitive to touch, in order to get to the primary somatosensory cortex, has to travel through the thalamus first. The same thing with your sense of vision. So this here is just an image of what I was kind of talking about a moment ago, when I had said, oh yeah, your left visual field goes to your right. Your left visual field goes to your left occipital lobe, but your right visual field goes to your right occipital lobe. So if we think about, actually, no, let's jump to this one for a second. So this is, let's just ignore this for a second. It doesn't exist. Okay. So looking at your left eye. Right. Here's your left eye. Cool. So this would be, yeah, okay, left eye, yeah, hello. So, temporal lobe. When we talk about the temporal half, so that would be kind of the left half of your left eye, right? And the nasal half, that would be kind of the right side of your left eye. Right, does that make sense, right? Like, if we talk about, right, this is your left eye. Anything that way is the left half, or left field. Anything that way is towards the nose, or it is right field. Cool. Alright. So just like before, how light, as it comes in, it crosses over, right? Same thing happens here. From the right visual field, so I'm just going to draw a line. Here's a right. This is all left eye. Here's left. So from the right visual field, cross over, and it actually synapses, or it stimulates the photoreceptors on the left half of the eye. Well, from the left visual field, crosses over, and it stimulates receptors on the right part of the eye. And that's where it kind of gets tricky or funny or whatever. Maybe it's already got tricky and funny, I don't know. Right, but we talk about the left visual field. It stimulates photoreceptors on the right half of the eye. So it actually continues and travels to the right occipital lobe. Right, so think about it. Whatever half of the eye it stimulates. Right, so my left, because it comes in diagonally. Right, so because of that, the left visual field comes down and contacts the right part of the eye, right? The right part of the retina. So because it's the right part, it goes back to the right occipital lobe. And same idea, the opposites for the right visual field, right? The right visual field here comes in, crosses over, hits the left part of the eye, or the left half, transmits back and into the left occipital lobe. Right now, alright, why are we getting this much detail? What do I care? That's part of where we talk about like loss of vision, right? Blindness, that sort of thing. There's so many different types of blindness. There's so many different reasons why people can become blind, right? Right. There's many different reasons as to why, right? So people might not be able to see the lateral portions of their visual fields, right? For instance, great example. The way that this works, right here, we can't really see it, but right here is where your pituitary gland would be. Let's say you have a pituitary tumor. Okay, and it's growing and growing and growing. Okay, it's growing. Awesome. One of the symptoms which can arise from that is a loss of your lateral, is basically a loss of your lateral vision or your peripheral vision. And the reason why that is because of the way that your vision travels back to your occipital lobes. I'm not going to get into crazy detail, but when we do this, right, for instance, if I look at, we'll just stick with this one because I've been looking at it. If this is your left eye, you can see your temporal half, right, so this is your lateral vision, comes in, hits the right half of the eye, comes in, and it crosses over. Well, that was your, that's your left eye, your temporal half. So that's the peripheral part, left, right, the outermost portion. That crosses over. Well, that's where there's a problem if somebody has, let's say, a pituitary tumor. Come on. Stupid pen thing. What's wrong with you? Or if somebody has like a pituitary tumor, for instance, it's gotten huge, gotten massive, it starts to impede on the ability of these neurons to cross over, hence the reason why they can start to lose their peripheral vision. Right? This occurs slowly, especially in people who have a pituitary tumor. Doesn't occur in everybody that has one. Nope, not at all. Right? But it's one of the potential symptoms that they can have. Right? And so it occurs slowly over the course of months. So oftentimes they don't even realize it's happening to them. Right? Literally. Right? They might notice one day, man, my neck's just beginning super stiff every time I look over my shoulder when I'm driving my car. And when you watch them, they're like, my God, it's because you're like having to spin the entire way around. Like, why can't you see better than that? Right? It occurs slowly, so they don't see it, or they don't. No pun intended. They just don't. Right? They just don't notice it. Right? So, anyways, that makes sense. Right? So, right, our sense of vision, right, definitely the most, right, complex per se, and it really has more to do with the sheer number of structures that are involved with it. There's definitely a number of structures involved with our sense of hearing as well. Not quite as many as with our sense of vision. But here I'm going to pause and upload this video, and then I'm going to create a new one, finishing up with auditory as well as some kind of a couple other things. So I'm going to put that second part up here in just a moment.